Great, so I'd like to thank the organizers and switch topics a little bit into the world of chemistry. These are my disclosures. So I'll start off with sort of a little bit of an introduction into kinase inhibitors, make sure it's up to speed on this topic. And then I'll talk about three projects, one that's published, one that's about to be published, and one that's not published, all related to cyclin-dependent kinase inhibitors. So the hope in the kinase inhibitor field was that we were going to find a Gleevec-like molecule for every tumor. And of course, that never happened. And I think the field right now is sort of suffering from two major problems. The first major problem is a lack of generality for kinase inhibitors. All right, so clearly we've seen very dramatic responses to kinase inhibitors, but in a very limited number of tumors. So that's sort of problem number one. And then problem number two is that, as Jeff was talking about, we get resistance very quickly. And so clearly there's still a lot of work to be done focusing on kinases that are mutationally activated, finding second-generation inhibitors, finding allosteric inhibitors, finding compounds with novel pharmacology. But fundamentally, we need to think about other ways of using kinase inhibitors other than simply going after oncogenic kinase targets. So Brad did a great introduction into targeting transcription. So let me just give you sort of the chemist's rendition of this. There was really a golden era of targeting transcription once people realized that nuclear hormone receptors were great drug targets. And so why are these such great drug targets? Well, reason one is that their natural ligands are small molecules, hormones, signaling molecules, estrogen, progesterone, androgens. And so they've naturally evolved to bind small molecules, right? So it's not that hard to understand that mimic those natural hormones and either acted as agonists or antagonists. Second of all, when you target these nuclear hormone receptors, you get a very specific effect on gene expression, often in a very specific tissue type. Then sort of fast forward to the 90s, once we started understanding the sequence specificity of transcription factors, there's a big wave of effort in the medicinal chemistry community and also in the academic community to try to find compounds that could specifically disrupt the ability of TFs to bind DNA, or even more ambitiously perhaps, find compounds that could interfere with key complexes that form and drive transcription. I think it's fair to say that those efforts really met with quite limited success. But as Brad also introduced, there's been now a realization that instead of sort of going after that very difficult medicinal chemistry challenge of DNA-protein interaction or small molecule protein-protein disruption, why don't we simply go after the post-translational modifications that we have experience with now and that are much more easily drugged. And so that's led now to an effort to target kinases, methylases, histone deacetylases, and a whole variety of enzymes that regulate this transcriptional process. And then the other motivation other than chemistry is really this notion that a lot of cancer is simply driven by deregulated transcription. And it's frustrating to think about this because, as molecular biologists and structural biologists, we like the idea of a mutant protein, a mutant inhibitor, hand in glove, thinking about the molecular interaction. But if a lot of tumors are simply being driven by rewiring of transcription, and again, Brad gave a beautiful example of where there was deregulation of the PDGF receptor, where there was a strong driving signal from PDGF receptor. I think that's actually the exceptional case. I think in many cases, what's happening is with transcriptional reprogramming, you're getting many small changes, and there's not a single molecular target necessarily that can correct that general transcriptional deregulation. So being a kinase-focused lab, we naturally started thinking about what are the transcriptional kinases that are well characterized. And the kinase that's been the most carefully characterized in this process is a kinase called cyclin-dependent kinase 9. Okay, so CDK kinases were initially made famous for their role in controlling the cell cycle. They were identified in yeast and other model organisms as the kinases that control cell cycle progression. But then years later, folks realized that there were certain kinases, like CDK9, that have a key role in turning on the ability of RNA polymerase II to transcribe and make mRNA. And the way this kinase works is RNA polymerase II, this is the enzyme that makes mRNA, has a long C-terminal tail of 52 repeats of a 7 amino acid sequence that gets heavily phosphorylated, initially characterized by CDK9, but now we realize CDK7, CDK12, 13, and possibly other CDKs are involved in that transcriptional regulation. And so we began to investigate different targets that could be looked at for inhibiting this transcriptional process. And there's been beautiful work by my former colleague Jay Bradner to make BRD4 inhibitors. Here's a transcriptional coactivator, generally important for transcription. You get very dramatic transcriptional repression when you inhibit BRD4 with compounds like JQ1 and others. Beautiful work I encourage you all to look at from Matt Scher on CDK8, inhibiting CDK8 with a natural product called cortistatin. But today I'll focus on our efforts to target CDK7 and CDK12. So one of the big challenges with CDK inhibitors is it's a 20 kinase family, about 20 CDKs, and the ATP site where most of these drugs bind is extremely homologous. And so it's been very hard for certain CDKs to get selectivity. There's some exceptions. For example, the CDK4/6 inhibitors that have been approved are relatively selective. But for other CDKs, it's been very challenging to get selectivity. And we had devised an approach to sort of systematically find scaffolds that are privileged for selectivity. And I'm not going to go into all the details, but it involves making libraries, looking at kinase selectivity profiling, finding compounds that have specific selectivity, and then picking scaffolds that are privileged for having selectivity towards a given kinase of interest. We've primarily focused on covalent inhibitors. This turns out to be an area that's really had a resurgence of interest in the pharmaceutical industry. There's many covalent inhibitors that probably everybody in this room have eaten at one point or another, but there has historically been a reluctance of the pharma industry to work on covalent inhibitors. That's eroded recently with the approval of several covalent EGFR and BTK inhibitors. And so these compounds, what they do is they form a very specific covalent linkage with non-conserved cysteine residues in and around the ATP binding site. And this turns out to be a really powerful way to get selectivity for a given kinase of interest. And the reason this works so well is that you have now two selectivity filters to get you selectivities. Right? Normally, you just rely on non-covalent recognition of the compound by an enzyme to get selectivity. But now, based on where the cysteine is in and around the active site, if you put the reactive group here, you can hit this cysteine. If the cysteine is over here, then there's a different way to get selectivity. So you have this way of breaking selectivity into two pieces. The other advantage is with kinase inhibitors, when you do pharmacological experiments, you're always wondering, is it on target or off target? Well, here, if you make an obligately covalent compound, you've got a built-in way to prove whether the compound is selective. You can simply make a resistant allele, mutate that cysteine, and ask, does it rescue the inhibitor pharmacology? Turns out there's a lot of kinases that have a cysteine in and around the active site. Almost a third of the kinome has a cysteine somewhere around the active site, and only about a dozen or so of these kinases have been annotated yet to have a selective inhibitor has been developed. So really a lot of meat on the bone for developing covalent inhibitors. So we were interested in targeting CDKs, and we wondered whether we could get selectivity with covalent inhibitors. And so to make a long story short, we performed a screen of a covalent kinase inhibitor library looking for compounds that could block transcription in a cellular phenotypic screen, and we stumbled across this compound called THZ1. Then we used a kinase profiling technology to determine that this compound was very selective as a CDK7 inhibitor and obligately required this reactive moiety to get covalence, and the corresponding non-covalent inhibitor didn't form this covalent bond. So we had this pair of compounds, covalent and non-covalent. But what was confusing was that we knew CDK7 did not have a cysteine in and around the ATP site. So why was this compound able to be covalent? Turns out there is a cysteine, but it's actually not in the kinase domain. So actually the C-terminal tail of the kinase snakes around over here, and there's a cysteine that's dangling here in the breeze by the ATP site. So what happens is the inhibitor binds, the arm of the inhibitor reaches to the outside, and is able to tether to this cysteine. And this shows you the advantage of doing a phenotypic screen. You would not have anticipated that kind of an inhibitory mechanism being possible. So we could then corroborate this compound very nicely inhibits CDK7, inhibits the RNA Pol2 activity. Here's a Western blot showing that. And the other key function of CDK7 is to regulate cell cycle transitions. And so it's what's called the CDK activating kinase. It phosphorylates the activation loop of the cell cycle kinases like CDK1 and 2. And you can see the compound also inhibits that phosphorylation. We then did this critical rescue experiment to prove to ourselves that the compound was having an on-target effect. So we mutated the cysteine, and we could show we could then restore phosphorylation of the CTD. This only rescued the anti-proliferative activity of this compound by about tenfold. So that gave us some indication that despite this compound looking quite selective based on the kinase profiling, it may have had some additional targets. We then collaborated with Cyril Bennis over in the Navy Yard to look at what this kind of a CDK7 inhibitor would look like. I thought, here's a key kinase regulating transcription and cell cycle. This is going to be an extremely cytotoxic compound. And surprisingly, there was a very wide range of sensitivities of the cells. So we had cells that were killed at 10 nanomolar, 100 nanomolar, 1 micromolar, and 10 micromolar. And we really didn't understand the physiological basis for that sensitivity. And so we started working with Rick Young, who had been collaborating at the time with Jay Bradner on a very similar question, analyzing this compound JQ1 that it works as a BRD4 inhibitor. And there they had a similar phenomena. Why does an inhibitor of a general transcriptional coactivator protein show selectivity? And they'd come up with a model that basically predicated that there were a subset of genes whose expression was hypersensitive to a transcriptional inhibitor based on being regulated by what they called a super enhancer. Okay? So these super enhancers were enhancers that simply had more of the transcriptional apparatus bound to them. And so if you look here at essentially the load of proteins bound at the enhancers, most enhancers have very little. And then, just like the wealth distribution in the US, you get to this point, and then all of a sudden, these genes have all the enhancers. And just like comes up in taxation discussions, these are the ones that are extremely sensitive to talks of taxation or to talks about transcriptional deregulation. It's a little bit counterintuitive because you might have thought these have the biggest enhancers, they're going to be the most robust to small changes in transcription. But if you actually look at the transcripts that crash out very quickly, and the ones that are regulated by super enhancers, you'll see a quite good correlation. And you'll then be able to construct typically some sort of a model as to why a particular cell type, so in this case we were looking at a TALL cell line, is extremely sensitive to this transcriptional inhibitor. So in this case, we're able to rapidly inhibit the sort of core transcriptional regulators, RUNX1, TAL, and MIB, in these cell lines and lead to a very strong apoptotic response. Surprisingly, this kind of a covalent CDK7 inhibitor was reasonably well tolerated, and you could actually see quite nice efficacy in disseminated xenograft models. So this sort of led to the idea that maybe there are cancer types where they're more sensitive to the need for continual transcription that would be sensitive to these types of transcriptional CDK7 inhibitors. And then in collaboration with a number of labs, and I don't have time to go into all the stories, they were able to find a number of tumor types that were hypersensitive to these CDK7 inhibitors. Some of them you might expect, for example, MYC-driven tumors. This was already known. These were very sensitive to tumors, for example, to inhibitors of transcription, CDK9 inhibitors, actinomycin D. They were also very sensitive to covalent CDK7 inhibitors. And then some that you wouldn't have expected. So, for example, triple-negative breast cancer, and some really elegant work done by Jean Zhao's lab, they were able to show that triple-negative sensitive and in contrast, not hormone-dependent breast cancer were much more sensitive to transcriptional inhibitors. And this sort of led to a simple model, very analogous to what Brad outlined, is that we have tumor types where you have clear oncogenic driver events, also like Jeff talked about with mutant EGFR, where you get a dramatic apoptotic induction upon inhibition of the driver oncogene. But then there are some tumors where typically we haven't found strong genetic drivers. Another example we found is, for example, small cell lung cancer. We know you lose p53, we know you lose RB, but you don't have a mutant oncogene kinase type event there. They tend to acquire the ability to proliferate through transcriptional reprogramming. And in general, we see these tumor types to be hypersensitive to these transcriptional inhibitors. And the therapeutic index that you can see, I'm talking a therapeutic index in terms of cell lines, that you can see with these transcriptional inhibitors will often be similar to what you see with an oncogenic kinase inhibitor. So you can get a 10 to 100-fold sensitization based on this transcriptional addiction. And so I think time will tell now, in terms of this transcriptional addiction, it's not, uniquely you have to go after CDK7. There's probably many other enzymes in this transcriptional regulatory complex that'll give you similar pharmacology, and the question really is going to be about the therapeutic index associated with this approach. So that leads me to the next player in this transcriptional complex that we started analyzing, a kinase called CDK12. And we really stumbled into this kinase serendipitously, trying to figure out why we weren't able to rescue all the inhibitor pharmacology from that initial compound, THZ531. So Tingyu Zhang in the lab realized that there were other kinases, CDK12 and 13, that also have a cysteine on this C-terminal loop that extends in front of the ATP binding site. So here's CDK7, here's the cysteine here, and in CDK12 and 13, there's another cysteine, cysteine 1039, that's very proximal to this ATP binding site. And then using a medicinal chemistry campaign, which I don't have time to go into, we were able to change the linker arm from an arm that could reach both CDK7 and CDK12 to a linker arm that was much more selective for going after the CDK12 or 13 cysteine to make probes that were selective for those kinases. So we used a pull-down approach to prove the compounds could label CDK12 and 13, but not CDK7. We could corroborate that by mass spectrometry, that we labeled the appropriate cysteine. And then in collaboration with the SGC, we obtained a crystal structure corroborating very nice covalent bond formation with the cysteine of CDK12 and then subsequently with Matthias Geyer on CDK13. We were able to make a biotinylated compound where we essentially put on a linker arm here and then a des-thiobiotin, and we could show very nicely that we can pull down CDK12 and 13 from cell lysates, and that we can have very specific activity of the active compound in terms of proliferation inhibition that requires not only the presence of this covalent bond, so that proves that the compound is covalent, but also the stereochemistry of this compound is critical. So the active enantiomer has this chirality. If you simply flip this chirality, you get a completely inactive compound. This is very important for pharmacology to be able to prove that the compound is on target to have an enantiomer that doesn't have the activity. We could show the compound is impervious to washout, which is what you'd expect with a covalent inhibitor. So when we did this project, much less was known about what CDK12 does, and so we did ChIP-seq, looking at where CDK12 lives across the genome, and we showed that it was coincident with marks of active transcription, where RNA Pol2 is and where H3K27 acetyl is, suggesting that this enzyme is present in the regions of the genome where there's active transcription ongoing. We then were able to show that unlike the original compound THZ1, which could inhibit all the 3 CTD phosphorylation marks, the effects of these covalent 12 inhibitors were most pronounced on serine 2, with much less effects on serine 5 and 7. The initial compound we made, THZ531, had very poor in vivo pharmacology, so we had to go in and make additional CDK inhibitors, and knowing that we had this strategy for reaching the cysteine, we simply went in and looked at other known CDK binding scaffolds and then installed this arm that could reach out to the cysteine of CDK12 and 13. We could corroborate these were very nice and potent 12 and 13 enzyme inhibitors. And then we devised a nice strategy. This was a clever idea that Nick in the lab came up with, which was to create cell lines where we specifically mutated of cysteine, either singly, CDK12 or 13 singly, or doubly. We made a cell line that had both the cysteine mutant in 12 and 13, and then could ask, both in terms of proliferation and signaling, what degree of effects are coming from 12 and 13, because our compounds can't separate those two enzymes. And without going into all the data, we could basically show that these mutant cell lines, when you take away the cysteine, they become impervious to pull-down labeling of the drug. And we could show a nice rescue of inhibitor pharmacology, right? Whenever you see a kinase inhibitor paper, always look for this experiment. Do they prove that the mechanism is on target? Have they shown some kind of a rescue phenotype? And this is what we're showing here, is that, here's the wild-type cells killed at 25 nanomolar. The 13 mutation does not affect proliferation very much. The major driver of the proliferation effect here is 12, and then if you make the double mutant, you get a quite nice over tenfold rescue of the proliferative effect, showing that you have a lot of on-target effect with this tool compound, MFH290. We again showed that if you go to a sufficiently high dose, similar to the CDK7 inhibitor, you see broad-based downregulation of transcription. But surprisingly, when you go to low dose, you get a very specific effect on a subset of genes. And when we went in and look at what those genes are, it was a huge number of genes involved in DNA metabolism and DNA repair. So this was a real conundrum, because here's again, a CDK kinase that lives at active enhancers known to phosphorylate RNA Pol II. Why is it having a very specific effect on a subset of genes? And here's some genes, for example, that it's affecting, BRCA1, 2, RAD51C, and you can show very nicely downregulation of these genes, and then you can show rescue by the inhibitor resistant mutants. And we actually don't have an answer to this right now. Our current working hypothesis is that there may be a non-RNA Pol II substrate of CDK12 that's involved in gene-specific regulation of these factors, and we're trying to do phosphoproteomics to figure out what that is. But it shows you, for example, that this very general transcriptional CDK has an additional role regulating DNA repair. Again, this is showing inhibition of the targets. You can see very strong downregulation of RAD51, and then you can see how nicely this gets rescued when you introduce that cysteine to serine mutant. So the question is this really functional? Is it just having an effect on gene expression, or is it really functional in cells? And here we're looking at a very simple assay of DNA damage, looking at RAD51 foci. And what you can see here is that the double mutant can really rescue the effects of MFH290, which are to enhance the degree of DNA damage. So this led us to an idea, really borrowing on the work of others on PARP inhibitors, is that would there really be a synergy with other agents that can damage DNA? And so here the idea was that if you had cells that were partially resistant to PARP inhibitors like olaparib, and you were able to inhibit CDK12, would you get synergy? And you actually get really impressive synergy. I mean, I've done a lot of combination studies. You don't often get synergy curves that give you these kind of incredible Bliss scores, but you can really get nice sensitization of DNA damage, really suggesting a new role for CDK12 in regulating the DNA damage response. We're just now figuring out the pharmacology of these compounds. What doses do we need to get in vivo target engagement? This is actually another advantage of covalent inhibitors, because the compound forms a covalent bond with the target. You can then do pull-down assays to establish whether you have target engagement. And in collaboration with Jean Zhao's lab and Jeff Shapiro now, we're just in the process of starting to do in vivo studies to corroborate this effect of looking at synergy with DNA damaging agents. So I really do think that CDK12 inhibition might be sort of a PARP type mechanism that will be active in relevant tumor types. So in my last little segment here, let me talk about a third approach, a third CDK, CDK9. So CDK9 actually was the transcriptional CDK that had been the most well characterized at this point. There's a famous drug called flavopiridol, which is a pan-CDK inhibitor, but does have very potent CDK9 inhibition, and so a lot of the pharmacology of flavopiridol has been ascribed to CDK9. However, I think a more up-to-date view of that is that that probably comes from many additional targets. So we really wanted to know, could we make a very selective CDK9 inhibitor? So the technology that we turn to for this approach is a new technology involving recruitment of small molecule binders of ligases. And just to give you a lightning introduction into this technology, this goes back many years to an infamous drug called thalidomide that caused a huge number of birth defects due to being a teratogen. People had no idea of its mechanism of action. Years later, folks here and elsewhere figured out that these compounds worked very well in myeloma. This was commercialized to make several imide-based drugs that have really become part of the combination therapy for multiple myeloma, still in the absence of a molecular mechanism. And then finally, beautiful work from a number of groups elucidated that actually these compounds, the imide-type compounds, bind to a class of E3 proteins, for example, cereblon, and bind to them. But that still, didn't provide a mechanism until beautiful work from Ben Ebert and from Bill Kalin and others showed that what these molecules essentially do is they act as molecular glue to bring and promote the degradation of some of the natural targets of this E3 ligase. And so what they do is they bind to cereblon, they act like a little piece of sticky bubble gum, and then they bring in the substrates for degradation. And so essentially, you make this ligase inappropriately degrade additional target proteins. And this led chemical biologists, and really the pioneer in this field has been Craig Crews, to readapt this approach to making bivalent compounds, where instead of just relying on this molecule to be a bubblegum for natural substrates, why don't we try to recruit unnatural substrates by making bivalent compounds? And so Craig calls these PROTACs. Other groups have called these different names. But basically, it's a bivalent molecule whose job it is not to act as an inhibitor, it's to act as a certain piece of molecular Velcro to degrade certain targets. And so the question is, how general is this approach, and can this be used to degrade new targets of interest? And so, as I mentioned, we were going to go after CDK9, and there was this compound known in the literature, developed by chemists at Bristol Myers Squibb, called SNS032, which was a very potent CDK9 inhibitor, but it does have a number of other targets. So a lot of CDK inhibitors are supposed to go after the CDK family, and then for some reason, a lot of them tend to cross-react with GSK3 alpha or beta. And so then we took this degrader approach. We made a bivalent compound, so we're linking the CDK binder to this imide degron moiety. And the first experiment we wanted to do is, how does it change the binding specificity of the compound? And because this binds to the kinase, this dangles in the breeze, the selectivity actually of these compounds in terms of binding kinases is pretty much identical. They bind very similarly to the two kinases. But amazingly, when you take this degrader and you do a whole proteome study, the only protein that we find being degraded is CDK9. I mean, this was just a stunning result to me. You have this compound that's promiscuous for binding to CDKs. It's only degrading CDK9. And this is not just by fancy proteomics. You can do the tried and true Western blotting. And look at this, at 1 micromolar, you get partial degradation, and then and this is actually another thing I should point out for these compounds, you actually get better degradation over a certain concentration range. If you go too low or too high, just like Goldilocks, you get in trouble. And the reason for that is these compounds exhibit what's called a hook effect, right? So there's a concentration range where you can bring the two partners together. If there's too much engagement on the left-hand side or the right-hand side, then you don't bring them together. And so there's basically this critical range. You get degradation. Here you get too high, and then we actually didn't go low enough for this compound. You get too low. The other thing is you have a very powerful way to prove these compounds are on target because you know they have to be ligase dependent, okay? So here we created a cell line where cereblon, the essential ligase, is knocked out by CRISPR. When you do that, you no longer degrade, okay? So you really can show this is on target to the ligase degradation mechanism. And then you can prove that it's proteasome dependent, and you can compete it off by either the kinase binding bit or the cereblon binding bit, you knock the PROTAC compound off, and then you rescue that, that degradation. And so the question is, why are we getting selectivity? Why aren't we simply degrading all the targets to which this compound binds? And to look at this, we use this very nice technology developed by Ben Cravath's lab called Kinative, where you can essentially measure the ability of an inhibitor to block certain targets from labeling. And so a little bit of a complicated experiment, so bear with me as I explain it. You basically can take live cells or lysate, you treat them with inhibitor, and then you have a reactive probe. And the probe will label kinases in the ATP binding site, but it'll only label targets to which the drug is not bound. Okay, so for example here, only in the inhibited sample do you protect GSK3 alpha from being labeled. So that proves indirectly that the drug is binding to GSK3 alpha and not to these other targets. But there's a further complication here, because what this measures is loss of signal. But with a degrader, you can lose signal either by binding or by degrading. Okay, so to discriminate between those two possibilities, you got to again use this control of looking at a cell line that has the ligase and a cell line that doesn't have the ligase. And so that's what we did. We looked at the kinative profiling in wild-type cells or in these null cells. And what you can see is we can only see degradation in the wild-type cells, as I showed earlier, but you actually can't detect, the drug concentration is so low that you can't even detect the target engagement. The only reason that we're seeing this loss of signal is because of degradation. And when a lot of chemists first looked at this technology, they're like, this is never going to work because these molecules break all of Lipinski's rules. They're too big, they're too hydrophobic, they're not going to get into cells. But it turns out the reason that you are allowed to break Lipinski's rules is because these don't work as classical pharmacological agents. Their job is not to get in there and have target occupancy the full time. Their job is to go in there like a square dance and do do-si-do with their partner and get the degradation to happen. And so in this case, you don't actually want really high target residency and target occupancy. You want catalytic turnover. And when we've gone in and tried to measure the concentration of this degrader compound, a typical kinase inhibitor might get into cells at 1 micromolar, 10 micromolar, or if it accumulates even 100 micromolar. These cells get in below 100 nanomolar, so we can actually have a hard time detecting intracellular drug concentrations. And that's actually what you need to get this degradation. So a lot of the selectivity seems to be because we're in a concentration range where the compound is only binding to a subset of its targets, right? So we always heard, like for example, a multi-target inhibitor like dasatinib. Yes, at 1 micromolar, dasatinib binds to 100 different kinases. But at 1 nanomolar, dasatinib is actually a very selective Src family inhibitor because its inherent affinity for Src family kinases is much higher than for other kinases. So what this PROTAC technology is allowing you to do is get into this really low concentration range where the inherent selectivity differences between targets is observable. So that's clearly one thing that's going on. However, other things are very likely also going on, although we don't have direct proof of it right now, related to the geometry and the trajectory for which this ubiquitination has to happen for the degradation, right? So for the degradation to happen, the substrate has to be recruited in a certain orientation. The appropriate lysine has to be sitting there. You've got to get the ubiquitin transfer. You then got to get the subsequent polyubiquitylation. And there's no doubt that certain trajectories and certain complexes will favor that more than others. So we then asked, what does this, degrader do? And of course, this was not too hard for us to follow up because there's been a lot of literature with CDK9 inhibitors. And so one thing that CDK9 inhibitors will do very effectively is deplete short-lived proteins like MCL1. And you see that, very nicely. You see very strong induction of caspases. You see PARP cleavage, as you would expect. And because this mechanism is irreversible because of protein degradation, it's a little bit similar to the covalent inhibitors I talked about earlier, right? So they're irreversible because the protein is permanently modified. The only way to restore enzyme function is through new protein synthesis. Likewise here, you're getting degradation. You can only recover function by new protein synthesis. And so you have essentially made a irreversible inhibitor, even though it's a reversible binder. So how do we compare this to a selective CDK9 inhibitor? And we went and looked in the literature, and it turns out chemists at Novartis had made a very nice covalent 9 inhibitor, which they somehow never bothered to publish, but it was in a patent application, and we synthesized it. We called it NVP2, and we profiled it. This is a pretty selective non-covalent CDK9 inhibitor, and we wanted to compare it now to this degrader compound. So if you bear with me here, so if you look at this NVP2 compound, it's killing cells at about 10 nanomolar, and it's just a regular inhibitor. So there's no difference between the red and the orange curve because it doesn't care about the ligase. It's just binding to CDK9, inhibiting CDK9 function, inducing apoptosis. Whereas with the degrader compound, the thal compound, you can compare this green curve and the black curve. So here you're getting 2 logs of difference in killing depending on whether the ligase is there. So again, showing that this compound that we've made is not very effective as an inhibitor. It's really acting as a degrader. We're obviously validating this strategy first with known pharmacological binding compounds, but the real promise of this technology is you can actually turn non-functional binders into degraders. And chemists have known for many years that you can often find binders to proteins that are non-functional. They've been typically put in the drawer because what are you going to do with those compounds? Now there's a way to repurpose some of those compounds by basically reprogramming them from being binders to being degraders. Here's the point that I made earlier about this being irreversible. Okay, so essentially looking at how much annexin positivity you can get with or without washout. So with a non-covalent inhibitor, you get good induction of apoptosis. You do washout, and of course the compound is gone. You don't induce apoptosis nearly as well. With the degrader, it's almost impervious to washout, very similar to a non-covalent compound. So let me wrap up here and just give some thoughts about kinase inhibitor therapy. So you often go now to meetings and you hear everybody talking about immunology, which I think is justified, but it does get a little frustrating when it's a non-immunologist talking about immunology. So I'm not going to pretend to do that. And I want to argue that actually for targeted therapy, it's actually incredible that we've seen the results that we've had with monotherapy, given all the resistance mechanisms that we've recently talked about. So I really think the route forward now is we have to have multiple pharmacological strategies on target to go after not only the oncogenic targeted protein, but also the collaborative pathways like I've talked about, transcriptional processes. There's a number of kinases now where we've been able to find compounds that bind to second sites. And so work we did earlier with Novartis, we developed allosteric ABL inhibitors that can bind here, and they can be combined with ATP competitive inhibitors, right? If you tie down the enzyme with 2-site binders, it's much harder to get resistance. Likewise, we've been doing this now with Michael Eck and Passiani for EGFR. We found a second site in EGFR right next to the ATP site. The ATP site is here. We found another pocket right here, very analogously to where the MEK1 and 2 inhibitors bind. Again, that provides 2 anchor points to keep the kinase downregulated. So really, talking to our friends in the antiviral world, they always ask me, why are you still doing, monotherapy with targeted inhibitors, right? So for HIV, we went from mono to double to quadruple now cocktail inhibitors very quickly. And monotherapy for HIV fails, incredibly quickly. Of course, the advantage that they had in HIV is these compounds can all be combined and they're relatively well tolerated because they go after viral proteins and not host proteins. So the challenge that we're really faced with now is we've been incredibly successful at developing all these different mechanistically distinct ways of going after cancer, right? And we know cancer co-opts all these pathways from metabolism, protein homeostasis, we talk about transcription, fooling the immune system, survival pathways, and signaling. And really the challenge right now is how do we come up with preclinical models to figure out what are effective combinations. It sounds like a very obvious thing to do, but having worked in cancer research now for a while, it's extremely hard, right, to make a model that faithfully recapitulates, one target. Now you want a model that faithfully recapitulates the clinically relevant setting for 2 different mechanisms, and that's, extremely hard to do. But I don't think we can do this randomly, in the clinic. Although it seems like some folks are trying with immunotherapy. You really need preclinical strategies for figuring out what are going to be the most effective ways of combining these modalities. So let me stop there and thank some really fantastic people who did all this work. So all the brains behind the biology and the CDK work was Nick Kwiatkowski, and in very close collaboration with Rick Young's lab. We had a lot of good collaborations with structural biology, Matthias Geyer, and the SGC. We've had a lot of help from the physician scientists at the Farber who have been really generous working with early stage chemistry. And I'll stop there and take some questions.
Nat, one of the things about flavopiridol is it's toxic, and CDKs are toxic. So I wonder, and we've done this with FKBP12 binders, if you can give a really nice toxic dose and rescue with thalidomide.
Yeah.
As a strategy.
Yeah, no, so I think the degraders, are a great way to explore intermittent dosing and being able to turn on and off the inhibitors in a way that's just not possible with regular pharmacology. So that's, yes, you can tune it. You turn it right off, the degradation right off, you give a dose of thalidomide. Yep.
Howard.
I'd like to know if you could expand on, you commented briefly on the challenge going forward of therapeutic index for the first compound that you mentioned, the CDK9, 12, 13. Mm-hmm. You gave a wonderful historic review. I worked on some of those drugs years ago, and the covalent multikinase inhibitors, some of them failed that you didn't put on on the path there on your slide, and some of the ones that you succeeded, I think, have really gone after more of an enrichment, like a companion diagnostic approach, which is one way to improve the therapeutic index. So is that part of the thinking going forward? Could you expand on that, how you're thinking about it? Because just to say that it worked in a triple-negative breast PDX model, I think you have more thoughts about rationale for that compared to what you've said so far.
Yeah. So. So there's two sides to that question. So one is, all compounds have, off targets, and clearly with the covalent inhibitors, very oftentimes, there can be toxicology not related to the target. Clearly with CDK inhibitors, there's going to be, on-target toxicology, and I think really with the CDK7 inhibitor, for example, in mice, they've been in our hands, better tolerated than some of the pure CDK9 inhibitors, and now we've made selective CDK12 inhibitors. They've actually been more toxic than the compounds that inhibit 7, 12, or 13. Whether mouse toxicology is going to be at all predictive, I think probably not. There's a lot of work also with these cell cycle kinase inhibitors, polo kinase inhibitors, aurora kinase inhibitors. Those also work, very nicely, both in PDX and in xenograft models. They ended up, often having bone marrow toxicity before having clinical efficacy in humans, and most of those, didn't go forward. So unfortunately, I think for the different CDKs, because their mechanisms are so different and there's such species specificity to their toxicology, we are not going to know how well tolerated they are. Flavopiridol, there's some experience in the clinic from that compound, but that compound has some very unusual pharmacology that make it extra, complicated. So basically, it has extremely high protein binding, and you basically have to do this loading dose, and then just beyond that loading dose, it then causes, massive tumor lysis if you go into a leukemic, type model. So to address your question about, triple-negative breast cancer, yes, you can show selectivity or, in a PDX, but, is the transcriptional addiction that we observe in that model actually true in patients? And the answer to that is we just don't know. Yeah.
And any comments on approaches to molecular or genomic IHC, proteomic ways of selecting the best patients, an enrichment or CDX approach for the 7-12 inhibitor?
Yeah, so that's a great question. So in small cell lung cancer, we've worked on, for example, some of the GEM models. So the GEM models, typically they have RB and p53 deleted, and then after a certain latency, they will get small cell tumors. And we do PDX studies with those inhibitors there. We get, like you see in the clinic, some mice responding and some not. And right now we're basically in the process of looking at the effects on super enhancers, the effects on transcription, to try to understand, is it what the drug is inhibiting, in those cells or not inhibiting, or is there, certain additional genetic lesions that make the inhibitor, work better or work less well. I think there's going to be clear ways of doing the stratification, but they're not clear right now.